Vor is a company really trying to change the lives of cancer patients in need. So let me talk to you a little bit about cancer to start with. And I think, firstly, you can read our disclaimer at your leisure. This is the same as what's on our website. So traditional drug development, aside from the sledgehammers of chemo or radiotherapy, was that, you know what, cancer cells are different to normal cells. And what if we could actually target cancer cells through particular antigens on their cell surface and really pick out those cancer cells and kill them preferentially to healthy cells? And that's a great concept. That was really the whole concept that drove targeted therapy in its development. The unfortunate thing is, for the vast majority of cancers, they look exactly like normal cells. They are derived from us. They're derived from self. And unfortunately, from a molecular biology standpoint, they also look identical with very similar cell surface markers. And so when you end up choosing targets, whichever target you generally choose, you end up getting a lot of cell attrition of normal healthy cells, which leads to this phenomenon called on-target toxicity. What we're doing at Vor is we're thinking about this problem a little differently. Instead of thinking about targets or thinking about binders or potency, we are thinking about healthy cells instead. And what we're doing is we're protecting healthy cells through genome engineering strategies and essentially making them immune to targeted therapies, thereby making these targets much more cancer-specific and being able to kill the cancer instead of normal cells. What that means in reality is that current targeted therapies are really agnostic as to which cells express particular targets. However, in Vor's approach, whether you're using targeted therapies like antibody-drug conjugates, T-cell directing antibodies, or even CAR-T modalities, that you'll be able to potentially use these therapies in a much more specific way, protecting healthy cells and eliminating cancer cells. And so let me talk to you about three different aspects of what we're doing at Vor today. The first aspect is called Vor33. It's our lead program. It's a hematopoietic stem cell product designed to be immune to CD33 targeted agents. And we're doing this by removing CD33 expression. And so to talk to you a little bit about what we're doing with this program, we're in the clinic already, and we're treating patients with advanced acute myeloid leukemia. So AML is a disease of the bone marrow. It's where there's uncontrolled growth of these AML blast cells within bone marrow that can spread to the periphery. And despite the advent of transplant over the last 50 years, AML is still an immense issue. Even those patients making their way to transplant, they're more likely than not to relapse of their cancer after their transplant. And so what we are doing is we are substituting the original stem cell transplant with Vor33. So Vor33 is generated from the same cells you'd use for a regular transplant, but it removes CD33 expression. So in our clinical trial, what we're doing is we're taking the same cells that would be used from an allogeneic healthy donor, processing these and giving them back to the patient as a stem cell transplant infusion. That's administered exactly like any regular stem cell transplant today as in a central line. What we're also doing in this clinical trial that's currently ongoing is that we are monitoring the patients for engraftment. Engraftment is the primary endpoint of the study. And what this really measures is that Vor33 acts like any regular stem cell transplant. So despite the manipulations we're doing to it, we hope to demonstrate that these cells reconstitute the blood system like any regular transplant. Now, this can be very easily measured through peripheral blood monitoring of neutrophils or platelets. And our primary endpoint is at day 28 following their stem cell transplant. Now, another novel thing we're doing in the study is we're dosing Mylotarg to these patients around day 60. Mylotarg is an antibody-drug conjugate against CD33. It also happens to be very myelotoxic, really through its mechanism, because it kills anything expressing CD33, including hematopoietic stem cells and their progeny. And so this has never been done before, where you're dosing Mylotarg in a post-transplant setting where normally you'd never even think about using Mylotarg because it would be toxic to the graft you've just provided to the patient. In this case, clinicians, patients, and the FDA were comfortable with us giving Mylotarg in this post-transplant setting, really with the hope that we're not going to see the typical cytopenic toxicities you would expect from Mylotarg, and you could potentially benefit from this post-transplant treatment to keep the patients either in remission longer or even target cures. So that's exactly what we're trying to do at Vor. We have a number of great sites who are actively enrolling in the study right now, and we're pegged to read out our first clinical data from this study in the second half of this year. So it's one thing to actually do a study, but firstly, you've got to make product, right? And so this is our manufacturing process. We start with the same cells that you would use for any regular stem cell transplant. This is an apheresis product from an HLA-matched donor. We then wash this product for platelets. We then separate out CD34 positive cells to isolate stem cells. By the way, the separation process also allows us to isolate T cells that could be used for subsequent CAR T therapy. I'll talk to you a little bit about that later. And then to these 34 positive cells, we end up doing a genome engineering step. It's simply an electroporation with a Cas9 enzyme and RNA guide complex. And that you allow these cells to rest and repair, and that becomes a stem cell product. There's no viral vectors because you're not introducing any new genetic material to these cells. And you're also not growing out these cells either, because if you grow out stem cells, they tend to lose their stemness. And so the manufacturing time is roughly about 48 hours, including release. It's about a 7 to 10 day total vein-to-vein time. We're doing this through a CDMO right now, but we're also fortunate enough to be building our own CGMP manufacturing facility in Cambridge, Massachusetts. In fact, it's on the floor right below where I sit in my office. And so we have 4 clean rooms that are designed to be multi-purpose. They can each handle vector or not. We have 2 clean rooms customized for hematopoietic stem cell manufacturing, as well as 2 clean rooms for CAR T, and it's a real luxury to have this facility, which should be ready very shortly. So let me tell you a bit about our second program called Vor33. So VCAR is a CAR T therapy. There's actually 2 separate programs here, one which is using autologously derived cells, and the other using allogeneically healthy donor-derived cells. So on the left here, what you see is something called Vor33 Auto. So this is an autologously derived cell therapy where you're taking cells from the patient, you're processing them and introducing a CAR construct against CD33, and then you're introducing this back into patients with relapsed refractory AML. This is in an ongoing study right now. We're expecting data sometime this year. It's an investigator-sponsored study involving the National Maradona Program, the NCI, and several leading academic centers. What we are doing at Vor in-house is creating another program that we call Vor33 Allo. Now, this is inherently different for 2 reasons. One is that we're using healthy donor cells. So these are cells from a matched HLA donor. In fact, in this case, we're treating patients who have relapsed after their transplant. And so we would go back to the original transplant donors and ask for a T cell donation. And from these cells, we are able to get much more stem-like, healthy T cells that we can then use to process a CAR T that is inherently matched to the patient because they've received stem cells from this very same donor anyway. And so we are expecting to file an IND in the first half of next year for this program, where we're expecting potentially even better efficacy than the autologous program because of the healthier cell phenotype. So to think about cell phenotype, we've looked at this very carefully, and we think rather than just, say, equipping a soldier with a gun and focusing on the gun, let's also think about the soldier. And in this case, the soldiers are young, healthy phenotype where we're expecting robust in vivo expansion and proliferation of these cells, and hopefully really good persistence of these cells over time. And what you're seeing is that in our manufacturing process, we're able to get very nice stem-like phenotype of cells in the vast majority of both CD4s and CD8s coming out of the process. Now, what we eventually want to do is this vision that you see right here. So if I think about myself, if I were an AML patient, what would I like? Well, firstly, I would like a next-generation stem cell transplant that allows my bone marrow to be treatment resistant to anything else I give it. And the next thing I'd want is programmed CAR T cells that could live for months or years in my system without causing toxicities that you'd normally expect, but a program to hunt down any sign of cancer for months to years to come. And we believe this Vor33 VCAR33 treatment system could be the answer to do that. So in this case, patients would be getting Vor33 as their next generation stem cell transplant, and then after a few weeks, receive VCAR33 as this CAR T, hopefully with these T cells lasting for months to years, hunting down any signs of AML, really aiming for durable remissions or cures for this disease. So lastly, let me wrap up with what we're thinking about next, because not only could this be transformative, but we don't want to rest on our laurels, and let's think about what else we could be doing to benefit patients. And one avenue is looking not just at CD33, but looking at targets beyond that, where we could actually do multiple edits, called multiplex editing, and couple these to multi-specific CAR Ts. And so here we're showing data on additional targets that we've been working on. So outside of CD33, there's targets like CD123, CLO1, EMR2, and then in contrast, there's a target called CD5 that's expressed on T cell tumors. And so by investigating additional targets with the Vor approach, we think we could benefit patients even more, where you'd worry less about phenomenon like antigen escape mechanisms or tumor heterogeneity, and expand the platform to look at conditions outside of myeloid disease. What I'm showing you in this slide are two different methodologies where we could use multiplex editing either with Cas9, sequential Cas9 edits, or base edits, where we can delete out more than one target at a time with great efficiency. But not just that, we're also looking at minimizing off-target edits and translocation rates, and you can see here that we have a number of different strategies to essentially eliminate measurable translocation rates, either through sequential Cas9 edits or through base editing techniques. And you can see on the right side, we can actually render these cells very protected from CAR Ts. On the far right, you see data where these are double edited knockout cells, and in the presence of two separate CAR Ts, one against CD33 and another against CLO1, you get very robust protection of these cells and their populations compared to either single edited cells or wild type cells. So in conclusion, Vor Biopharma is a little bit of a different cell therapy company where our chosen modality is engineered hematopoietic stem cells. We're also working on complementary CAR T cells, really with the aim of long-term remissions or cures in AML to start with, and then going beyond. We have the luxury of building out our own in-house GMP manufacturing suite, and we have a great management team. And with our IPO early last year, we have cash into Q4 2023. I want to thank you very much for your time.